If you rely on diffusion, basically a random movement of molecules, it can only go about 0.1 millimeters, given the concentration gradient, of course. But when we're working the body, most molecules, they don't diffuse further than 0.1 millimeters. And so the cells need to make sure they're that close to a capillary. And so the density of this capillary bed is going to vary. It's going to vary depending on how metabolically active this tissue is. So it's directly related. So capillary bed density is correlated to the metabolic rate of tissue. And so the idea is this, basically. If you have some metabolically active tissue, let's say it's neurons, or let's say this is muscle tissue, you're going to have a higher density, you're going to have more dense capillary beds. And so what does a more dense capillary bed look like? Well, it means per volume of space, you're going to have more capillaries. So if I want to make this capillary bed more dense, I would just kind of draw more interconnecting capillaries here. This is me making capillary bed more dense. Making the capillary bed more dense means cells are exposed to more blood, essentially. There are multiple places and multiple surfaces that they can interface with to get access to a capillary. So more surface area means more exchange. Right, so what does a less dense capillary bed look like? So here's the arteriole, here's the vein, and so maybe you could look something like this, right? This is less dense than that. And so the density of the capillary bed is going to vary. How many capillaries you're going to have per volume of space is going to vary depending on how metabolically active that tissue is. And this is also what we call plastic phenomena. This means this can change. We can have something called angiogenesis, where we can have creation of new blood vessels to kind of create more capillaries when needed, or to break down capillaries when we don't need them. So, for instance, if you decided to do marathon training, right? Your leg muscles that sure need a lot of blood to walk around, but now even need more blood to run that marathon, you're going to start seeing some angiogenesis around those kind of leg muscles. So, generally there are two types of capillaries. We have something called continuous capillaries, and we have something called fenestrated capillaries. They're pretty similar, they're just some slight differences. For continuous capillaries, you have these leaky junctions that let in polar molecules that don't normally pass through a cell membrane. So we're talking about ions here, we're talking about glucose here. Small proteins can pass through here. These leaky junctions allow diffusion to occur better. Now remember, gases like oxygen and carbon dioxide, they can pass through, they're small enough to pass through cell membranes, so they don't need to pass through leaky junctions. They can just pass through membranes whenever they want. But for things like ions, glucose, they need some kind of transport, and one way is just to diffuse through these leaky junctions. Fenestrated capillaries are the same, but these just have larger openings. And generally you find fenestrated capillaries at the kidneys and the small intestines, places where you're going to have large amount of transport. So when you're expecting to move a lot of molecules from the lumen to interstitial fluid or vice versa, you're going to probably see fenestrated capillaries. And that makes sense, right? Small intestines absorbing all these nutrients needs to do it really quickly, relatively quickly, and so it will have these kind of bigger openings to allow more content to diffuse quickly. Now both of these types of capillaries can do transcytosis. Hopefully you guys remember what transcytosis is. Transcytosis is basically a concept that you take content from one side, kind of have the cell swallow it via endocytosis, have it transport across the cell, and then get dumped out onto the other side via exocytosis. That's what transcytosis is, right? It's basically just taking content from one side of the cell and dumping it to the other side of the cell via vesicle transport. Now, a neat thing that capillaries can do is if they make two vesicles and merge these two vesicles together, they can make a channel, a temporary channel. They're so thin that two vesicles can combine to basically make a temporary opening in these capillaries. So this is another way where you can create more openings for diffusion to occur. And this helps let larger molecules pass through when they can't make it past the leaky junctions. Does that make sense? Cool. Yes?
Is the basement membrane obstructed the diffusion?
The basic membrane? Not really. The basement membrane's pretty thin and loose on these capillaries, so you're not really expecting that much kind of interference. It provides some interference, but not a whole bunch. You can go into the basement membrane here in purple right here, but it's not that big of a deal. It's loose enough that these molecules can pass by with these. Alright, moving on to capillary bulk flow. Now I said molecules diffuse through, but when we're talking about bulk flow, we're talking about the mass movement of water. Okay. So water is actually being moved around by non-diffusion forces here. And so in this drawing, fluid's coming in this way, it's leaving out this way. And because of that, we can generally say that there is a higher pressure at the beginning, and there's a lower pressure, relatively lower pressure here. Does everyone know why I can say that? What principle allows me to say this? Yeah.
Probably because it's actually closer to the artery.
Well, this side's closer to the artery. This side's closer to the vein. But
doesn't have to do with the resistance?
exactly, right? There's resistance, there's friction here. So as blood flows along here, there's friction. It kind of like dissipates pressure. So that's why I can say relative on like the proximal end, the side closer to the arteries, there's going to be a higher pressure than the other side compared to the other side, which is going to have a lower pressure. And so this pressure is a hydrostatic pressure, and pressure is so high that it actually kind of forces fluid to leave the capillary. You can kind of think of a hose that has high pressure, and if you prick holes into it with a needle, it's going to start spewing out water from those holes you made, right? This is going to be something similar. These capillaries are going to have these holes, and because of this pressure, it's just kind of pushing fluid out because of the hydrostatic pressure inside the capillary. And so generally, what you see is there's a hydrostatic pressure pushing water out, and that's about at a 32 millimeter of mercury pressure. But that dissipates, right? Due to friction, that dissipates, and that dissipates to be about 15 millimeters of mercury here. So as we're moving the capillary, water is being kind of being pushed out of the capillaries due to this hydrostatic pressure, but that hydrostatic pressure is decaying as you go down the capillary. This is kind of representing the decay in the hydrostatic pressure. Everyone still with me? Okay. But there is a counterforce to that. So it turns out there is a hydrostatic pressure that pushes water out, but there's an osmotic pressure pulling water in. Sorry, let me draw that smaller. Yeah. It's about 25 millimeters of mercury. And that's because there are these proteins here that are osmolytes. They're osmotically active. They're going to attract water to them via osmosis. And so there's an osmotic pressure that brings water back into the capillaries. And for the sake of this class, it's the same throughout. It's the same throughout the capillaries. Also 25 millimeters of mercury over here too. So if you look at the difference between the hydrostatic pressure, so let me just draw the pressure line here. It looks something like that. It's just a straight line. And so if you look at this region of that region, that is signifying that more, in the orange region, that is signifying there's more water loss in this section than there's water gain in the capillary. More water is being pushed out of the capillary than it is being brought into the capillary via osmotic pressure. This is also called colloid pressure, the act of bringing water in. Now if you look at the other side here, black here, we're just getting Halloween colors on here. This side, in this part of the capillary, water is on net average coming into the capillary. Does that make sense? So in this region here, we have water gain, and in this region over here, we have water loss. And I'm talking relative. This is all